sequencing. So does anybody have any questions about these before we start? Okay. All right, so what I'm going to do is I'm going to start out with just a little bit of like biological background, so why you'd want to do chip-seq, what kind of experiments you'd want to do with it. We're going to next move on to the data processing steps, followed by some considerations to take into account with experimental design. Because I know that in addition to analyzing the data, you probably are thinking about how do I actually design a good chip-seq experiment, or how do I do this well in order to get clean data. And of course, if you design your experiment poorly, you're going to, no matter how much data analysis you do, it's going to be hard to rescue that experiment. So we're going to talk about some of those as well. We're going to talk about some analysis tools, and then, as mentioned, we're going to move on to this tutorial. So, first of all, y'all know what DNA is here. And I guess previously we focused kind of on the RNA, so these parts of the DNA that are transcribed into the genes. And you can measure the abundance of genes and the expressions of genes, and that kind of gives you an idea for what cell state is. The thing that I want to talk about today is not the RNA, not the expression of the genes, but the things that actually control the expression of the genes, which is lumped into this bucket of epigenetics. And so here we have the DNA, which is wound tightly around nucleus or histone proteins, as shown here. And each of these histones can actually have marks on it that kind of give you an idea of the context of the regulatory status of that particular part of the genome. And so then that gives you ideas or can give you an idea of whether this is an active region or a repressed region. And so just to kind of give you an idea of how complex this is, there's something that's kind of referred to as the Histone Code, which has kind of gone in and out of style over the years. But basically saying that if you could understand what modifications are made to the histones, then you could actually understand exactly what that part of the genome is doing. And so these are the histone proteins here. There's H4, so two H2 complexes, or subunits, H2A and H2B, as well as H4 and H3. And then the DNA is wound around these proteins. And they each have a very long tail that can have modifications, and some of the modifications are listed here. So there's modifications that signify an active part of the genome, as well as modifications that signify a repressed part of the genome. And so there's and labs are actively pursuing new marks for alternate states of regulation. But these are just a couple of them. And so we're going to focus on a couple of these, and I think generally if you're doing a chip-seq experiment, or if you're interested in histone modifications, you're only interested in probably about a handful. And the majority of these are on your H3 protein, or H3 subunit here. And so these are going to be H3K27, methylation and acetylation, for example, and H3K4 methylation. There's a trimethyl or single methylated. Anybody have any questions so far? So just know that there's like some activating marks, and there's some repressive marks. The most important repressive mark also is also on H3, which is this red line here, H3K27. So this is kind of the mark of repressed expression state. It's also important to note that these histone modifications can be read and written. So here's our nucleosome here again, and these are the tails here, and there's different proteins that will read and write these histone modifications. And so if you're studying a system where you know that there's some kind of gene that's upregulated or downregulated, it can definitely have an effect on the histone, the code, I guess, or the chromatin state. So the question that we're going to answer today is, okay, this is important to figure out the regulatory impact of any part of the genome, but then the question is, how do we read that out? So how many of you are familiar with chip-seq then? All right, so what do you guys know about chip-seq so far? How does it work? Sorry, what's your name?
Stephanie.
Stephanie?
Yes.
How does it work? What do you guys know already about this?
You have a protein that's somehow affiliated with your DNA, whatever your favorite flavor is, and you use an antibody, and you target that protein, you pull it down, and then you sequence the DNA that's attached to said protein.
Yeah, that's great. So basically, like Stephanie, sorry, what was it?
Yeah.
Stephanie said. So this is our DNA here. We have a bunch of proteins that are interesting. They could be a transcription factor. something like that, we cross-link it to our DNA so it's now stuck. And we're going to digest our DNA and create these little fragments. We're going to use an antibody against either the transcription factor or else against our chromatin modification of choice. And we're going to pull out just the pieces of DNA that are attached to this protein. So if we're interested in this yellow octagon, or hexagon here, sorry, we can use an antibody against that yellow hexagon to pull out the pieces of DNA that are associated with that. And then we're left, after we remove the antibodies, then we're left with the pieces of DNA that we know were associated with this protein at the end. And then if we sequence these, we can figure out, okay, what pieces of DNA had my protein of interest bound to it. Does that make sense? And so there's these 4 by 6 steps. So first of all, there's cross-linking. Next, we're going to shear the chromatin. Next, we're going to retrieve the protein or the histone modification using the antibody. And then next, we're going to release the DNA and then sequence it. Make sense? Okay. So, and I guess the reason, so this is a chip-seq, sorry. In the olden days, you used to do chip-chip, for example, which is where if you don't, you're able to, if you have a microarray, you can hybridize the DNA against some probes. But in the era of cheap next-generation sequencing, they've kind of moved away from this, and now you can actually just sequence the whole, I guess, all of the places that's bound by your DNA fragments. Sorry, I forgot to mention that. So these chip-seq targets can be, they can or chip-seq can be used to identify several things. So first of all, chromatin-associated proteins. So if you can do chip-seq against Hall 2A, for example, if you want to know regions that are being actively transcribed. You can do it against histone modifications, these, I guess, the Histone Code to figure out what the epigenetic context of each region is. You can also use chip-seq to figure out where your transcription factors are binding, and then also cofactors that are binding to the DNA, or they may not be directly binding to the DNA. And so when you do chip-seq, it usually will look like this. So this is, you're all familiar with the integrated genome, IGV, integrated genome browser, viewer, I guess what it's called. And so this is a IGV track for FOXA1, which is a transcription factor. And so here we can see this gene here. And what we see, first of all, is we see that there's like kind of a pileup in specific locations in the genome. And so the first step is to determine these enrichments. And then the next step is to call peaks. And so here you can see that they've called peaks. And so these are the FOXA1 peaks. And these are these extended blue lines here. I guess there's two sets. There's big peaks and then sub-peaks. So this big peak was called as a big peak, and this peak was called a big peak too. And so those are kind of the steps that we're going to be going through. But this is just kind of to tell you how it looks. So you'll have some peaks, and they're pretty clearly delineated because you're only pulling down the DNA that is bound at that point. Make sense? So this is a transcription factor. If you do histone marks, these are going to look slightly different. So this is H3K4 trimethyl. This is an active mark here, and you'll see that there are the same signal here. So you'll see that. Sorry, this is our treatment. This is our control. So this is in the treatment. We see this big pileup, and we see lots of reads that are aligning to this specific location, as well as these other locations here. We also have some reads that are kind of just aligning throughout the genome. We don't really care about those. We really care about these regions right here. We can call peaks on those right here as well. And you'll notice that these actually line up really well with the promoters of these genes here. So this is this gene right here, and in the promoter we have an enrichment of H3K4, which means that this gene is probably being expressed, as well as this gene right here and this gene right here. So you can see it's actually really clean, and it's really, really nice. However, not all histone marks are as nice as H3K4. Some marks look like this. This is H3K36, repressive mark. And you'll see that it's a lot harder to call these peaks. So this is your background, this is your control, and then this is your signal. So you can see that it's a little bit more complicated to call these broad histone marks. And we're going to talk about some of the challenges that are associated with either narrow or broad histone marks in a couple minutes here. Here you can see that it also coincides with the beginning of this gene right here, meaning that this gene is probably repressed. All right, any questions on the background? Great. So next we're going to talk about data processing. And I want to, not to scare you, but I want to start out with so are you familiar with ENCODE? So ENCODE is this big encyclopedia of epigenetic modifications, and they have a standard pipeline for processing all chip-seq data. And this is their pipeline. So basically what you're going to do is you're going to start out with some next, so remember we pulled down these pieces of DNA that were associated with our protein of interest, and we sequence them. That produces it's called a FASTQ file, right? Are you familiar with FASTQ files? Yes. So the FASTQ files will come in here. This is what's produced by our experiment. The first step is going to be mapping. So you already talked about next generation sequencing mapping, right? So we're going to map to figure out where all these pieces of DNA go in the genome. And there's a specific mapper called BWA, which is the ENCODE approved way of mapping reads to the genome. And then that will produce this BAM, which is this aligned file. Then what we do is they're going to create some statistics to figure out how well things align, and then they're going to filter the reads. So here they're going to get rid of unmapped reads that map to multiple locations, reads that have low quality, and duplicates, and they're going to throw those out. And they're going to then create a bed file, and then a compressed bed file. And from that, they're going to create some signal tracks like we were looking at. So asking where is the signal? And then you can also do peak calling, which are these two things we saw on the IGV track. So you want to know where is my signal, and what does it look like, and then also where are the peaks. And then from this, you can get correlation between replicates, and then you get this format called Big Wig, which is this compressed format to show you where the signal is. And then also, you're going to get peaks, which are going to be in a bed file. And then you can do this IDR, which is a technique that's used to determine how reproducible are my peaks. And you can take into account multiple samples or simulated samples to determine how likely is it that this peak that I'm seeing is a peak. And then they can also, after that, then they can do some motif discovery to say, okay, what kind of motif does my protein of interest like to bind to? And then from that, they can call motifs. Yeah. So this is the comprehensive ENCODE pipeline, and I'm going to take you through a couple of these steps. But it is somewhat complicated. At each of these individual steps, there are specific parameters that they've tuned. But the good thing is that there's actually a pipeline that they've kind of already made public, and you can kind of just run your samples through it. So if you want to have data of similar quality to ENCODE, you can use that pipeline, and I'm going to talk about that in a couple minutes here. But it doesn't have to be this complicated. So first of all, if we start up at the top left, we have these FASTQ files. You remember you've looked at FASTQ files, right? So they have the sequence of the DNA here. You have the quality. This line shows you the quality of each of the base calls, and then an identifier for each line. And from this, we're going to first ask, what is the quality of our data? And from this, we're going to use FastQC. And we talked about FastQC already, right? And so this shows us, for example, that this is a failed run right here, where after about 25 base pairs, our sequencing quality goes down a lot. But FastQC is going to be something that we're going to be using in the data analysis. The next step that I talked about is alignment, where we take all these short fragments and we align them back to the genome. Now there's a couple of, I guess, commonly used aligners in chip-seq data. The BWA is a very common one. It's the one that's used in the ENCODE pipeline. Bowtie 1 and 2, probably Bowtie 2 is also very useful, an alternate alignment method. And then STAR is another aligner that you can use to align reads to the genome. I believe actually for the second version, so for some of the ENCODE pipelines, they do use Bowtie 2, but you should achieve similar results using either of these aligners. Now importantly, after you align reads to the genome, you need to remove duplicates. So if you can imagine a situation in which if you pull down if you have this DNA that you know is bound to your protein of interest, and then you do some PCR, you are going to amplify. You could possibly amplify specific fragments more than other fragments, and that would make it look like there was a peak when there's actually not a peak in that location. So an important step, especially in chip-seq data, is to remove duplicates. And the way we're going to identify these duplicates is to find reads that have the same sequence and that they align to the same location in the genome. And then we're going to remove these, so that we're going to hopefully remove any PCR bias that may have been present in our sample. Other sequencing technologies will try to fragment the DNA into approximately the same size fragments. That's not always possible it's not always possible with chip-seq, so this deduplication step is very important in chip-seq. The next step in our processing pipeline is peak calling. So after we've aligned the reads to the genome, we want to say, okay, where are the peaks? And so this, there are basically two steps in this. First of all, the first step is you want to find a background distribution of tags. So you want to say, okay, is my pull down very specific, or is it kind of noisy? Is what I'm seeing at this location, is it more than what I expect to see in the background? Is it less than what I would expect to see in the background? But in order to know that, we have to define what the background is. So we come up with the background signal. And then second, for each location in the genome, I ask the question, do I see more tags, more reads, at this location than in the background? And then if I do, then I'm going to call that a peak. So that makes sense? So this is, there's a bunch of peak callers, and they all try to answer these two questions in fancy ways. Just to give you an idea of how many, a couple different commonly used peak callers. There's here's a couple of different peak callers, and you can see that they all try to model the background noise in a slightly different way. So this is a statistical model where they try to say, okay, which of the regions of my genome are background, which of the regions are signal, and they have a different statistical method for determining background, and they have a couple different assumptions. And so I've had good success with a couple of these tools. SciStar used Mosaics also, but the most common is Max, and this is also the one that's part of the encode pipeline here. And so there's a second version of this, Max 2, which is, I guess generally if you use Max 2 and you're processing a chip-seq data, you're pretty safe. It's very commonly used and accepted as a good peak caller. Although you'll probably get pretty similar results if you use any of the peak callers. So I'm going to talk for just a couple minutes about exactly how Max works, specifically Max 2. So Max 2, basically what it does is, I forgot to mention, there's one important thing with chip-seq. So if you imagine that you have your DNA, you imagine your DNA always runs from 5 to 3 in the 5' to 3' direction, and you have a transcription factor sitting on one of the strands, right? And so because when you sequence with your next generation sequencing, you have an adapter that goes on the 5' end and a 3' end, you actually, so you have a read 1 adapter that goes on the 5' end and a read 2 adapter that goes on the 3' end. What you actually do is if you look at the raw pileup of the reads, you actually see that there's a gap. So there's a gap between the read 1 and read 2. And so here they're calling them Watson tags and Crick tags, which is meaning for the read 1 and read 2. And so what you actually see is that the read 1s are all lining up on this side, pointing this way, and the reads 2 are all lining up in this location, pointing this way. And our transcription factor is actually sitting right in the middle there, because we can't actually sequence anything that's underneath our transcription factor. We only sequence the ends of the reads. Does this make sense? So if you were to pull up just the raw aligned reads, you would see a bunch of stuff that looks like this, where you'd have a double peak, and then nothing, and then another double peak. And then all that means is that if you were to look at it, you would see that, oh, my transcription factor is actually just sitting down right in the middle of these peaks. And the reason I'm not sequencing reads from that region is because my transcription factor is there. Does that make sense? So if you ever see this in your data in IGV, it's because you need to do a little more processing, and you're looking at the raw read pileup. So what Max does is it tries to look for these regions, and it says, okay, what is the distance between these two peaks? And from that, we can estimate the fragment size. So remember that, I guess in our process, experimental process, we digest the genome, right, to a specific length, right? And so if we digest it to 1 kb long, then we expect to see that all of our read 1s are piling up 1 kb from our other, the read 2s. Sorry, is this making sense? Do you have questions, confusion? So basically what it does is, first step is to determine what the fragment length was. And then if we can determine what the fragment length was, then we can say, okay, our protein is sitting, or our transcription factor is sitting right in the middle. Right in the middle there. Sorry, there's actually this picture right here that I should have been pointing to here. So we have the sense tags here, anti-sense tags, and this is the D, which is the distance between the pileups, which is the fragment length. Make sense? So now what we've done, that we've determined our D, is we're going to, given this sonication size, this bandwidth, this length, and a high confidence fold enrichment, this is the parameter you're going to provide it. You say, how much do I want this peak to be enriched over background? Max is going to take a window. It's going to slide a window of length 2 times whatever this bandwidth. So if I figure out that my digestion or my sonication size is 1 kb, we're going to take a 2 kb window. I'm going to slide it across the region, or across the genome, the entire genome, and I'm going to ask, do I have more than m-fold of tags than in the background? So if in this 2 kb region I have 200 tags, for example, and in my background I'd only have 10 tags, then I know that the fold enrichment is greater than mine. If I set m-fold to be 2, then I know I have twice as many tags in this region as in the whole genome, and so I call it a peak. And so this is the way that the max works. So basically it defines this bandwidth, it defines an m-fold, and it just slides this window along and asks, do I see more tags in this region than I would expect? Does that make sense? And then it calls those peaks. And so specifically the way it does this is it estimates this d, and then it shifts all these reads actually towards the middle. So it tries to figure out, it tries to put the reads actually under the transcription factor. And then it'll determine the significant peaks using this parameter based on the local read density. And so it actually is going to take into account the read density of the entire background of the reads. So the density of reads in the 1 kb background, the 5 kb background, and 10 kb background. This is a little more complicated. Do any of you want more details on how Max2 works? If so, I will explain this formula. If not, you can just say, you can just understand that it's going to try and find these windows where there's more signal, more tags than in the background. And there's a couple, okay, so then once Max runs, you can run it with a couple different modes. First of all, you can run with broad peaks. And so this will be, for example, these broad histone marks we're looking at. And so it'll actually look for specifically broad marks. And so this is a flag that you'll pass to Max2. Otherwise, it will just run in default mode. It will just look for these narrow, sharp peaks. And these are good for if you're doing transcription factor binding or transcription factor binding sites. And then Max2 will also spit out, in addition to the location of the peaks, it'll spit out the summit. So if you want to know the exact center of the peak, where it thinks that the transcription factor is binding, the middle of it, it'll spit that out too. And so we're going to look at some example output from Max2 in the tutorial today, later. Okay, any questions for how these peak collars work? So I guess just like to review some pitfalls. If you are looking at your data in IGV and you see these double peaks, that's because that's not signal. That's because it needs to be further processed, and you're actually just seeing the reads piling up on both sides of your transcription factor of choice. So they're not 2 different peaks. The peak is actually in the middle, and that's why these peak collars are useful. All right. Some considerations for experimental design then. Sorry, any questions, first of all? Yeah.
I do have a question. So is it like dependent on how you prepare your chip-seq sample if you get those 2 peaks? Because shouldn't you get, like, you're digesting out the protein and reversing crosslinks before you put the tags on, so your fragments are, 150-ish base pairs, so shouldn't you have them staggered all the way across whatever came down with your transcription factor?
Sorry, so you're saying,
Over there.
yeah, so you're saying, so what happens, look at this, a little more details, all right. I'll do a little animation next time. So basically what you're saying is here's your whole genome, and here's your transcription factor binding here, right?
Yeah.
And you're saying when you sonicate it, you can actually break in a couple places. You could break here, right? You could break here, right?
Yeah.
Is that what you're saying?
Uh-huh.
And this is actually what happens, yes. So if I am actually just
But the protein being there is going to protect it from the sonication.
Right. Sonication will just happen randomly.
Right.
But the important part is we're going to take our antibody and we're only going to pull down the pieces of DNA that were attached to it, right?
Right.
And then what happens is we're going to use our paired end read sequencing, right? So read 1, it's not going to sequence this whole read.
No, right, but sorry. So when you have so you sonicate, you'll get so you have this whole piece. And so you'll sonicate around your transcription factor, but you could have pieces of all these lengths, right? Because now this is in your precipitated thing. So then you don't have any tags on here yet, right? And so there's a part after this where you reverse your crosslinks and you remove this. So even if this is a slightly larger piece than the others, you still have it's still there, right? So so you'll have your 5' index here and here and here and here and here, right?
Yeah.
So why don't you get reads
all the way across this, or is it just that the transcription factor footprint is so large that you get a bias here?
Yeah, so sorry, let me back up a step here. So I think, sorry, I'm drawing a little bit
No, that's okay.
smaller scale than you are. So in sonication here, I actually, if you look at the same piece of DNA, it's going to look like this, right? Where I break right here, I break right here, I break right here.
Oh, okay, I see what you're saying.
And so, I mean, if there's ways to do, it's called exoseq, I think, where you actually digest using nuclease. You digest this to get an even shorter read. But I guess traditional chip-seq will just use the sonication or something like this to create larger fragments, so you don't lose them. But there are ways to actually digest even farther to get light around exactly where the root is.
Okay.
But so what we did is we basically have these long fragments, and we only pull down the ones. So in our pull down, we're going to not pull down this one because it didn't have an antibody against it, right? And we're not going to pull down that one because it didn't have an antibody.
No, right, I get that.
Just this bit is the one that stays, and then our 5. So then we'll have one left right here, one left right here, and this will only sequence 150 base pairs. This will only sequence 150 base pairs. So I guess there's a case in which if you did happen to sonicate right by the transcription factor, you'd sequence 150 base pairs out.
Right.
But you're also going to see 150 base pairs. But if you take all of these reads, then you're going to see something else like this.
Okay.
That makes sense?
Yeah.
Yeah.
Okay.
Does that answer your question, or does that make sense?
I'll read about it. It's fine.
If you want, you can give it another shot. If not, we
Yeah, no, I see what you're saying, but the biology doesn't make sense to me, so I have to read about why.
Biology doesn't make sense for why you wouldn't get reads at the location, or why you wouldn't get reads. Sorry, what?
I'll read about it. That's fine. Yeah. Thank you.
All right. So we're going to talk about experimental design really quickly. So things to keep in mind when you're designing your experiment in order to have a good chip-seq outcome. So most importantly, a clean chip depends on a good IP. So if you have an IP that's non-specific, or if you have an IP that doesn't bind very strongly to your protein of interest, you're going to get bad signal. And you're just going to get basically super ambiguous peaks, reads all over your genome, and that's not going to be a good thing. It's going to be hard to call peaks on these. However, if you have a strong IP that binds exclusively to your transcription factor, strongly to your transcription factor, you're going to get a strong IP. You're going to get a strong chip-seq readout. And so there's some suggestions that have been made. If you go onto the ENCODE website, they have a whole list of like 20 things that they require before you actually submit your data to ENCODE to make sure you validated your IP first of all. So if you're running in your own chip-seq experiment, make sure you're using a validated antibody, or that you validate your own antibody. Make sure you validate the enrichment before sequencing. You do this using PCR or Western. So if you run your so this is important if you're doing this for the first time, for example. And the challenge behind this is that there's no way to predict a priori the number of peaks or the number of locations that are bound by your protein of interest. So if I know that if I were to know that my transcription factor only bound 5 places in the genome, it would be easy to tell whether my IP was working without sequencing. However, if my TF binds every 5 bases, it's going to be hard to tell whether I have a bad IP or whether it's just going all over the place. And so this is a tricky thing in designing a chip-seq experiment. Generally, for transcription factors, we assume they don't bind everywhere in the genome; that they only bind specific regulatory regions. But especially for histone marks, it's hard to tell whether my IP is bad or whether this mark is just everywhere in the genome. It's important to have controls. So a chip-seq peak should be compared against the same region in a control sample. And controls can be different things. So these can be, there's a couple different controls. So you can do what's commonly called your whole cell extract, which will be the WCE, or the input DNA. So if you're browsing Geo, or you're browsing some online database for libraries that have been, or next generation sequencing library, like read data, and you see something that says WCE, this is the control that they use for their experiment, or the input is another word that they use to describe their control. And basically what this means is you do your sonication or your fragmentation of your genome but then you don't pull down anything, and you just sequence that DNA. That's the whole cell extract. Mock IP is, I guess, slightly more, is slightly different in that you're going to do your IP, but you don't have your antibody in there. So you technically shouldn't be pulling anything down because you don't have any antibodies. You can do a non-specific IP, which is an antibody against some protein that's not bound to DNA, for example, IgG. And so if you pull down everything that's bound, or if you pull down, so basically you should pull down any specific fragments of DNA. However, there's no really consensus on which control is the best. So feel free to use any of these controls. Just make sure you have a control for your cell type. Yeah, so controls should be generated for each cell type and for each antibody and each fragmentation strategy. So if you have your control done using sonication, don't use that control with something that you've done with like exoseqs or something like that. Make sure your control is specific for your cell type and your antibody. And this is important because your fragmentation sonication, so sonication breaks places that are open chromatin basically, that are nucleosome depleted. And so in your cell type, your cell type may have open chromatin at a region where another cell type may not have open chromatin. And so it's important to have this control for your specific cell type. However, if you have different time points in the same cell line, you don't need a control for each time point. Or if you have multiple conditions, multiple treatment conditions, you don't need to have a separate control for each treatment condition. So it's important to have a control. And then another consideration is sequencing depth. So we talked again about some transcription factors could be very specific in their binding, only binding 4 or 5 places in the genome. And for these narrow sharp peaks, you don't actually need to sequence that deeply to figure out exactly every region where your transcription factor is binding. However, if you are sequencing something like H3 K27, for example, or some other like broad histone mark, you'll have to sequence a lot more depth in order to get all the locations where it's binding. So this is just something you should think about. How deep do I need to sequence? And this will be informed hopefully by the type of experiment you're doing, as well as some knowledge hopefully you have about your protein of interest. All right, any questions? Okay. So with these, I think with these considerations, they'll help produce a more clean experimental outcome. Sorry, we're doing, all right. So I want to wrap up. Well, the last part of this is just a couple tools that I've either used or found to be useful or look like they could be potentially useful, and some kind of different use cases for these tools. So first of all, there's a lot. So I think that the type of tool that's becoming more popular is the type where you upload your FASTQs and it'll kind of do all the analysis in a standard pipeline for you. And one of these is called chip-seq, and this is it's an open platform for data analysis. You basically download it, and then you can run a bunch of analyses and visualizations through it. This is these are all in the slides. So if you want to go back and use any of these things, you can do it. But they do a bunch of you can do like heat maps, you can do visualizations, you can do scatter plots of binding through these things, and then it also shows you a workflow of what you actually did with your data. There's a program called EA-seq, which takes in, you have to have mapped data. So after you've already mapped your data, you upload it here. And this is basically they compared it, their tool against a bunch of other things, and they say that their tool does a whole bunch of things, including it'll test for signal strength in your data sets, it'll calculate logical differences, it'll do nonlinear normalization, it'll do a whole bunch of things. And so it seems like a pretty comprehensive toolkit to visualize and analyze chip-seq data. And then these are a couple types of the plots that it produces. And these are, are you familiar with this type of plot right here? So this is basically saying, this is a region. So for example, these could be all promoters, and each individual row represents a different promoter. And you're saying the intensity of the color represents the intensity of the binding or the enrichment of the reads of that region. And so here you can see that this protein of interest was enriched at these promoters, for example. And then if you were to look at this other protein, you can see that it was enriched in a slightly different set of promoters. You can also have some plots that look like this, where on one axis you have one treatment condition, another treatment condition, and see that generally peaks are enriched or differentially enriched in different samples. So these are kind of the plots that are produced by this. Next is Galaxy. So Galaxy is this, again, have you used Galaxy then before? So Galaxy is this comprehensive end-to-end solution where you can upload your FASTQs, and then there's actually a chip-seq pipeline that you can actually run in Galaxy. It's where you upload your FASTQs and it'll run everything. So it's actually in this, if you go to the Galaxy page, it's next generation, let's see. Yeah, so actually in this, there's a whole set of tools for peak calling, and in here you'll find Max. Max is this tool that we were talking about that's kind of generally accepted. So here you're going to put in the experiment name, what type of sequencing it was, and then you can upload files for your experiment as well as your control. You tell it basically, in order to calculate the background, it needs to know how big is your genome. And you can leave this as it's 2.7 billion for humans. Tell it, you can estimate your tag size, your bandwidth or fragmentation size, and then it'll run Max2 for you. This is really useful. So this is Galaxy. Do you do your tutorial in Galaxy?
No, not in this class. I do that in the one-day course.
Yeah, sounds good. But this is in case you don't have access to a cluster or a computation framework. Galaxy is a great collection of pipelines that can be used to analyze data. I mentioned previously Encode. Encode is a standardized, or Encode is this huge repository of, it has a bunch of chip-seq data in there. And if you actually go on GitHub, this website right here, you can see that you can have access to the pipeline that they use. And so what they've done, I've run these before, it's pretty simple to get up and running. If you're interested in running it here at MGH on the cluster, you can let me know and I've kind of set it up to run here. But you can run it on the Google Cloud, you can run their pipeline on the DNASys platform, you can run it on a couple different supercomputing platforms using, for example, CERM or SGE or LSF. I don't think it's listed here, but that's the one that we use here at Partners. And so they've implemented their pipeline on a bunch of different things. And so you can, if you want to get results exactly like Encode did, I highly recommend doing it like this. Then there's a lot of tools for analyzing the peaks and trying to figure out informative data from the peaks that you call. And one of these is called MEME, the MEME data set. And so MEME is kind of a larger project for motif finding that has a bunch of tools. The tool that's specifically developed for chip-seq is called MEME chip, which will go through and it'll find motifs that are enriched in your chips. And just kind of a broader overview of the MEME, it's a suite of tools. So there's MEME, if you just give it a bunch of sequences, it'll find common motifs in those sequences. Dream will find motifs that are similar to yours, or known transcription factors that are similar to yours. There's a whole bunch of tools as part of this MEME suite. But if you want to specifically look at MEME chip, you basically tell it the mode you want it to run in, you upload your peaks locations, you tell it what kind of motifs you want to look for, and then you tell it go, and it'll produce something that looks like this. And so here we see that in our peaks that we called, the most common motif was this that had a bunch of G's in it. Are you familiar with this PWM? Yes? Basically the height of each letter tells you the information or the likelihood of that letter being, that nucleotide being found at that position in the motif. So here you can see that this G is strongly conserved at these 3 bases as well as base 6. However, these small letters tell you that there's not actually, the motif is actually, there's not as much specificity at these positions 10 and 11 down here. So that's my first motif it found. It found it using this program called MEME, and then it finds that KLF1 and KLF9 are similar motifs to this motif. And then here it shows me near each of my peaks, it was actually found in the center of each peak, which gives me high confidence that this is actually has some biological information, or it's biologically informative. And then if you want, you can also find, you can also do some motif spacing analysis from this page. And then of course it'll list a bunch of motifs that were significant. So this is my second motif, my third motif, and so on. So this is a really cool, useful tool for if you have a whole bunch of peaks and you want to know what, like if you don't know the binding motif of your transcription factor and you want to find it out, meme is a great way to, great place to go for that. Kind of competing with meme is a suite of tools developed at MIT called Homer. Homer, I've actually not had as much success with this, but they claim to kind of do the chip-seq processing kind of for you once you got aligned reads. So they'll take the aligned reads, they'll call their own peaks, not using max, but kind of using their own Homer proprietary, or I guess their unique peak caller, and then they'll do a motif analysis on that. Do you actually use meme or Homer or max?
I actually, when I used to do chip-seq, I used meme.
Okay.
I don't
But
like
for the
Homer.
tutorial, do meme, or you'll.
For this tutorial, we were actually not gonna do meme, but
Okay.
sure, I can,
You don't have to.
it could be a homework.
Okay, sorry. I mean, it's actually not that difficult. All you do is literally upload your peaks,
Yeah, and then
it'll run.
Yeah, it's easy enough to run it online.
Yeah. All right, but Homer spits out a similar information as meme, where it'll tell you, it'll give you this this motif, and it'll say the same types of things. So here we see the 3 T's are highly conserved in our motif, kind of gets less specific towards the end. It'll tell you the p-value of the motif, how unique is this motif compared to any random sets of peaks, and then kind of some, if there are matches to known motifs, it'll tell you that. I'll give you the whole list there as well. So this is Homer, and again, Homer can be, on each of these slides I've put the web address for these things. So this is at Homer, UCSD.edu. It was here at MIT. At any rate, yeah, it answers the question also. So what motifs are enriched in my peaks, and also it does peak calling annotation, like I said. So as your, so the main output from chip-seq is a bunch of peaks, and of course you'd kind of, another question you could have is, what peak, do my peaks overlap with any other regions of interest? And so in order to do peak arithmetic, there's a really cool tool called bed tools. How many are familiar with bed tools? Yeah, okay, I won't spend as much time on this. But basically, if you want to, it's super useful in chip-seq because you can answer questions like, what peaks overlap in 2 experiments? Which peaks are unique to 1 experiment? And then also, which peaks are closest to genes? These are all questions you can answer using bed tools. And so, just one tool in here is bed tool subtract. For example, if you have, these are peaks in your normal sample, and then if you apply some kind of drug and get these peaks, you want to know, okay, what are my new peaks? And so you'll subtract the old peaks from the new peaks and get this. And this is the command line that you can do for that. By the way, I've also, in the bottom right of each corner, I'm showing a little command symbol for whether this tool is a command line tool or a web tool. So this is a command line tool, although you can access it on Galaxy as well as if you want. So then another question you may want to ask is, are peaks bound more frequently in one experiment versus another experiment? And this is a really hard question because if you have 2 experiments and you see a bigger peak in one experiment than in a smaller peak in another experiment at the same location, it could be the case that you just sequence more deeply in one experiment than the other experiment, or it could be the case that there was less background in one than the other. And so it's actually a very hard, somewhat unopened or unanswered question for how do I tell if there's differential binding between 2 different chip-seq experiments. Could it have just been the case that my Sonicator was a little more, like the voltage on my Sonicator was just different in one experiment that produced a different result. So this is it's a very, I guess, hard to answer question. Diff-bind is kind of one of the commonly accepted statistical frameworks for answering this question. So I refer you to diff-bind for definitively answering, is there differential binding in these peaks? And so this is based on some of these tools you may recognize from RNA-seq. So dseq2 or edger are ways to kind of try and take into account variability that may arise from experimental noise, for example, and to try and find true signal from that. And so dseq2 is implemented in R, and it's also available on Galaxy as well as in command line. And it'll produce these type of plots for you. For example, this is a heat map showing these are each of my individual peaks, and this is the intensity shows you how much was that peak bound in each of these samples. So this is diffbind. Another awesome tool that I use, it's an online tool called Great. And this tool, basically, if you want to know what are the characteristics of my peaks that came out from chip-seq, all you do is you go to this website, which is listed down here. You upload, I guess you choose which genome you're going to have, and then you're uploading your region, this bed file, and then you can upload some background regions, or you can just leave it whole genome, and you tell it go, and it'll tell you how many of your regions were promoters, what is the average distance to a promoter, what is the average distance to known disease-located, associated locations in the genome. So Great is a super useful, intuitive, and fast way to get some, I guess, biological interpretation of your peaks after you've done chip-seq. My tool of choice for visualization is actually Deep Tools. Deep Tools is a semi-statistically rigorous approach to measuring and comparing enrichment between different samples. So basically what you do is you align your reads, as the first step was, and then you will pass those aligned, you'll create a big WIG file, so enrichment track from your aligned files, and you'll pass those into Deep Tools. And what Deep Tools will do is it'll go across the genome and ask, okay, what is my enrichment signal at each location in the genome? And from that you can create really nice plots like this. So this shows, for example, these are transcription start sites and transcription end sites. So these are genes right here. And there's 3 kb upstream and 3 kb downstream of the gene. And so if you imagine all my 20,000 genes are in this location right here. And the color intensity shows either lack of signal or strong signal. And so you can see that in my H3K27 trimethyl input, in this cluster I had binding at the start of my transcription start site in this cluster. And by the way, this is all automatic, so it automatically, if you tell it I want some clusters, I want 4 clusters, it'll cluster all of your genes into 4 clusters with different characteristics as well. And so basically, so these are you can compare across different samples, you can compare across different treatment conditions. So this is really, really useful, and it'll produce these plots for you without a bunch of headache for you. So I highly recommend Deep Tools. To produce this, you need 2 steps. The first is called compute matrix, and for this you're going to pass in these big wigs. These are the signal tracks, as well as a region. So this I passed in my region for my genes for HG19, and then I want to say I wanted 3 kb upstream, 3 kb downstream, and I want my regions all normalized to 5 kb long, and then it'll produce this matrix, and then I just say plot heat map, and it makes a nice heat map for me. Yeah, with that, I think that's all the tools I wanted to cover. Are there any questions you have about other tools or other applications of chip-seq or analysis strategies? Sorry, that was like a little bit long. Nothing? All right. Yeah, so do you want to do a little break, or do you want to do any questions? Or
No, let's do a break for 5 minutes, and then we'll get back.